Here we go. Gonna wait for the bell. Hold on. Okay. So, Cell Hall of Fame. Now that we have the internet and instantaneous information, we don't spend a lot of time looking back at history. And we would not be where we're at today in science without the founding fathers or the people behind us. So we need to take a step back and think about how we know what we know so that we're not just taking it for granted. So, your first question asks you to fill in the missing events in the timeline that they have here as you're reading. I think it continues on to the next page also. And so, as you're reading, fill in this timeline of the beginning of how we knew we were made of cells and what a cell is. So, your explain question here, when it's asking you to think outside the box, it wants you to write a paragraph. And you can put this paragraph on the last page of this lesson where you have like the graph paper looking stuff. That's a good place for you to put this paragraph. And then you do have to know what the cell theory is and its parts. So make sure that you write that down, put a star by it, and understand what each of the parts mean. So early technology was very primitive. Um, First, we started using glass and bending it for optics. And so that had to start in the works of science at the same time that we were studying things. And we had magnifying glasses. And so then we started to put those together. And so we started seeing that the things we had in our everyday life were made up of smaller things. And so Robert Hooke is one of the first scientists to really encapsulate the term cell. And so he took a piece of a wine cork and sliced it really, really thin and looked at it underneath a microscope. And what he saw were these little cubicle like things, and he called those cells. Like the cells in, I think he visited a, what do they call it where the priests are? Is it I don't know. Can't think of it right off the top of my head. So you can see how primitive the microscope is over on the left, in which he had the eyepiece that he looked in, and then he used the little magnifying glasses in between him and the specimen. So really primitive. And we're probably going to watch this video in class together, or I'll try to link it for you. So we're going to take a minute and watch this. Well, no, we're not. We'll watch it in class together. But this shows some of the things that they would have seen with the early microscopes. So this is a sample of pond water, kind of like if you were swimming in Lake Lanier. This would be some of the stuff that would be in the water. So we'll watch this together. I'm pretty sure. I'll try to link it also for you. So Anton von Leeuwenhoek um, was the first to start describing these things that he saw in the pond water as animalcules. So CULES, kind of describing something small. So small animals, things that we could see only with magnification. So he called them animalcules. And there's some drawings, some of his drawings. Then we have Sliden, and he had the idea that plants are all made of cells. And then Swan stated that all animals are made of cells. And so you put all this together and you get the cell theory. One, all living things are made of cells. Two, cells are the basic structure. And three, all cells came from other cells. It's kind of like the chicken from the egg. Did the cell come first, or did the offspring cell come first? You know, that sort of question. So these three things are the cell theory. So make sure you know the cell theory. It's on the test. Promise, cross my heart. Girl Scout song. And so these are just some readings that come from another textbook that go along with what we're covering. And so you can pause these and do the readings if you want to. Remember, you can pause. Up, up, up. Oh my goodness. So here will be another video I'll put up for you. And again, it's pond. And these would be the kind of things that if you swallow some lake water, you might be swallowing. No worries though. Most of them won't harm you. Some other pictures of the electron microscopes. These are so exciting. Um, these weren't really around when I was in school, and the detail that we can see is just amazing. Um, I took my students to see the transmission electron microscope at the University of Arkansas, and it took up a whole room. And so it was crazy. And so we can see the difference between the two here, a 3D image versus a 2D image. So just amazing stuff that we can see that we couldn't see before. And the next slide is even more exciting. So this is a paramecium in three different types of telescopes. So you have the light microscope. That would be something we could look at here at school. But that's pretty close up. It's probably not the ones that we have in our class. Then you have the paramecium in the middle with a scanning electron microscope. Remember that the colors are something that the computer assigned. And then on the left, we can see a transmission electron micrograph. Each one gives us different pieces of information. And so we might use the TEM to look at the internal structures and the SEM to look at the external structures. So again, this is just amazing stuff. Amazing. So we can't see a lot of the small stuff with our naked eye. Our eyes aren't that developed, but we can use magnifications and microscopes to help us look at things. So here at about the 1 millimeter, that's about the diameter of one of your hairs to give you an idea of size. When we're getting down here to nanometers, that is like a meter with 9 zeros before it. So like 0.00000. Okay, really, really, really small. So I'll give you some comparisons here. Mhm. So to give you some comparisons of sizes. Okay, this is something that you've definitely heard before. So again, this is a part where you can stop and do the readings, take some little notes in your textbook guided note pages, and pause as you need. So if you don't already have a comparison chart in your notes somewhere, you should make one. And this is a good gift for that, made by the Amoeba Sisters. So you definitely are going to have to be able to compare and contrast a prokaryotic cell with a eukaryotic cell again. And if you're not already thinking about this, keeping them classified into their different kingdoms is also something you can't get rid of. That's information that you're going to have to hold on to. So make sure you jot this down into your notes too, unless you remember it, and then you're golden. So prokaryotic cells are very simple. Animal and plant cells are more complex. Some differences between the animal cell and the plant cell. You can see a rigid cell wall in the plant cell. The animal cell doesn't have this cell wall. It's made of cellulose and chitin. Not chitin, that's animals. But the cellulose, we can't digest cellulose. So if you eat paper, you're not digesting the cellulose that's within those plant cells. The plant cell does have a cell membrane, like the animal cell does. It's just underneath the cell wall. The cell wall allows for several protections that the animal cell doesn't have, and we'll be talking about that probably when we get to osmosis and diffusion. You notice in the middle of the plant cell you have that large kidney bean, jelly bean looking blue thing. Well, that's the central vacuole, and that is a place that allows the plant cell to absorb and store water when needed. And it all has to do with osmosis and pressure. And hopefully we'll get to that. So if you haven't already got this, I'm sure you do. Um, this is something that you will need to be able to do. And so I'm pretty sure you guys probably already have this in your notes since we did most of that in the last lesson or the last unit. We were probably going to watch this together. Again, review of the cell theory. You can pause these slides as needed. It's the same thing that we had before. I just highlighted some of the important stuff. And looky there, just for you, just for my good kiddaroos. Move this over here so you can pause it here if you want to, and move it back. Well, I can be anywhere. Oh, I don't want to be there. That's it for that one.